Now, why would you want an alpha subunit that actually promotes growth? Well, we would argue that this is perhaps because of its selective expression in vascular endothelial cells. In situations of oxygen deprivation, there is a population you would like to continue to proliferate, and that is the vasculature in a so-called neoangiogenic response, and this is something we're currently investigating. But as we've heard, Myc is a potent regulator of anabolic metabolism and cell division through its myriad of target genes, and Myc is deregulated in many human cancers, either by heightened growth factor signaling, amplifications, translocations, even in some cases, point mutations in the coding domain of the Myc locus. But we can show that Myc, even if expressed at low levels, can have an important role in at least one very HIF-driven tumor, and that is renal clear cell carcinoma. To remind everyone, renal clear cell carcinoma is largely driven by VHL mutations, so the alpha subunits accumulate throughout these tumors regardless of the relationship to blood vessels. And 30% of these, we've actually looked at hundreds of patient samples at this time, exclusively express the HIF-2 alpha subunit. And in this preview that Bill Kalin wrote, he and us have argued that this flavor of renal clear cell carcinoma, which exclusively expresses HIF-2 alpha, are very dependent on the connection between HIF-2 and its positive effects on c-Myc in multiple ways. Now, the 60% that co-express both HIF-1 and HIF-2, and we've actually found they're almost at stoichiometric levels, this means they're neutral for effects on c-Myc. So these tumors are not c-Myc driven, and instead appear to be dependent on, for example, deregulated EGF receptor signaling or heightened mTOR activity. And this would suggest to us that these patients could now be stratified for targeted therapies. For example, the HIF-1, HIF-2 tumors would be better candidates for things like rapamycin analogs, sorafenib, sunitinib, and perhaps the HIF-2 expressing tumors would resort to something else like perhaps anti-angiogenics. But Myc is also a member of a multi-protein family, and whereas c-Myc is broadly deregulated in a number of human cancers, n-Myc is overexpressed more selectively in tumors of the central nervous system, including neuroblastoma, and L-Myc is instead detected in small cell lung cancer. And we were interested in seeing if we could extend the connection between HIFs and Myc to another family member, i.e. n-Myc, and began using neuroblastoma as a model system for the following reasons. So neuroblastoma is the most frequent solid tumor in infants and children, accounting for approximately 10% of all pediatric deaths. The primary tumors usually arise in places like the adrenal medulla, but can also by PET scan be detected in areas of the neck, chest, and pelvic region. Metastases occur to bone, bone marrow, liver, and lymph nodes. And this was a very attractive model to us because of the interest in this important clinical problem, but also through collaborations with our colleagues at Children's Hospital, John Maris and Michael Hogarty, we have access to a large number of carefully cataloged patient samples. And so we wanted to, oh, and before I get to that, I should mention that John in particular has done a beautiful job of showing that mutations in ALK kinase or polymorphisms in LMO1 or BARD1 can clearly contribute to this disease. However, n-Myc amplification remains the most significant prognostic indicator for how this disease is going to progress. n-Myc amplification, which leads to very high levels of n-Myc expression, can be found in 20 to 25% of all the tumors, but actually 40% of the high-risk categories. And as shown here, high levels of n-Myc usually indicate a very poor outcome for these pediatric patients. So can we play the same game? Is there a distribution of labor with the HIFs? Are they expressed differentially in the different categories of neuroblastoma? And so we sort of played the same trick. We acquired a number of patient samples, and we found something that was surprising to us, that the n-Myc single copy, which now instead express c-Myc exclusively, is the tumor type that expresses HIF-2. And again, there's a collaboration between HIF-2 and c-Myc in the NMYC single-copy neuroblastomas. But in multiple MYC-amplified neuroblastomas, instead of expressing any HIF-2, they instead express HIF-1. We saw this by both immunohistochemistry as well as RNA expression profiling of over 100 primary tumor samples, and this just indicates that the NMYC-amplified high-risk tumors maintain HIF-1 expression, but they completely lack HIF-2 activity. So, as I indicated previously, when MYC levels, either NMYC or CMYC, are at very low levels in the cell, that HIF, in part, can inhibit their activity to slow rates of, for example, cell proliferation, again, to conserve ATP and promote cell survival. But she has argued that if MYC is overexpressed due to mass action, it can actually escape this inhibitory effects that HIF-1 might provide. And in fact, we see this in one example of an NMYC-amplified tumor. When they're placed under low O2, rather than slowing cell cycle progression and exhibiting decreased rates of entry into S phase, they maintain it. And under either normoxia or hypoxia, these cells remain completely dependent on continued NMYC activity. Now, we've shown, which may not be so surprising, that these, of course, exhibit what we've been calling the Warburg effect, over 1.5 molecules of lactate produced for every molecule of glucose consumed. And even at baseline levels, different enzymes in the glycolytic pathway, like hexokinase 2 or LDHA, if we interfere with HIF-1 activity by RNAi, their levels decline. If we interfere with NMYC, their levels decline. And in two cases, if we combine HIF and NMYC inhibition, levels decline to extremely low levels. So going back to the primary tumor samples, we showed that a number of glycolytic enzymes are overexpressed in these tumors, and one that was very highly expressed in the NMYC-amplified samples was lactate dehydrogenase, and has been clearly described previously. Just like other MYC-transformed cells, these NMYC overexpressing neuroblastoma cells are completely dependent on lactate dehydrogenase for in vitro growth, and in vivo, if lactate dehydrogenase is inhibited, these tumors grow very poorly, if at all.